Hello, everyone, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and I'm here with Aki Ko, CEO of Elixirgen Therapeutics, a clinical stage RNA therapeutics company focused on the discovery and development of cell and gene therapies. They are advancing 3 technologies to develop therapeutics for a broad spectrum of diseases, including rare and genetic diseases and age-related disorders. Aki, welcome to Cell and Gene, the podcast. So happy you're here today.
Thank you for having me.
Good. Okay. So I just gave a pretty broad explanation of Elixirgen Therapeutics. And so I would love for you to explain it to us in your words and talk us through a little bit about the company's mission.
Yes. So I think that was a great introduction, and I will further add that overall, we are a RNA technology company that is obsessed with patient safety. And then one gut check that we do for ourselves is that if it was me, would I be okay with receiving this therapy? And we believe that with creativity and with strong core technologies, we don't need to trade off safety and efficacy. So our commitment to the patient safety and comfort is a very high priority for us. And in that same vein, we develop therapies with the end product and its application in mind from the very beginning. And despite being a small company, we've at this point developed a ex vivo cell therapy using an RNA virus with no DNA phase that is currently in phase 1/2, a self-replicating RNA vaccine that has completed a phase 1/2 study, and now this brand new design for linear mRNA called Bobcat mRNA that is in the preclinical phase.
Good. Okay. Thank you. And thanks for talking about the gut check as members of the company and how you talk about patient safety and how would I want to be treated? That's such a really thoughtful way to go about the development of these therapies. And I'm glad you brought up Bobcat mRNA. That's what I want to talk about next. Talk to us a little bit about it. So again, that's Bobcat mRNA, the therapy, and then the why behind it.
Yes, of course. So I think what and why. So first, what is it? Bobcat mRNA is a proprietary 5' and 3' UTR design that can encode large proteins over 13 kilobase while remaining compatible with existing RNA technologies such as modified nucleotides. It can be manufactured at high quality and produces proteins at levels comparable to that of standard mRNA. And it's called Bobcat because it does not need a poly A tail to produce proteins, similarly to how a bobcat has a short or no tail. The why behind it is that this capability for encoding and expressing large proteins unlocks a variety of potential treatments for diseases that currently have a high unmet need, particularly those with a large gene that's involved.
Good. Okay. Thank you for that. All right. Now, especially when it comes to treating Duchenne muscular dystrophy or DMD, I want to talk a little bit about that. So how does Bobcat mRNA differ from traditional exon-skipping drugs and AAV, microdystrophin in treating Duchenne muscular dystrophy? And I guess a follow-up question to that is specific, regarding delivery, what is the advantage to the mRNA approach over the AAV approach?
Yes. Thank you, Erin. And I think I'd like to start with the patients first. And the first is that while there is so much active development for patients with DMD, I think there's still a highly unmet need, particularly for the non-ambulatory folks and for the older population as well, where most of the existing therapies are targeting still ambulatory and much younger pediatric patients. To go into the point for Bobcat and full-length dystrophin is that first, a full-length dystrophin is highly desired for the treatment of Duchenne muscular dystrophy, and it has not yet been delivered. The biggest issue with using the full-length dystrophin is the DMD gene's very large size. It's the largest gene in the human genome, and its protein cannot be packaged. It cannot be packaged within an AAV, which only holds roughly 4.5 kilobase. The current gene therapy approaches that are in the clinic right now use an AAV encoding a micro or mini dystrophin that uses up to a 4 kilobase truncated DMD protein, because it's been shown that microdystrophin delivery could result in slower progressing muscular dystrophy, similar to Becker muscular dystrophy, which is in which a truncated dystrophin is expressed, and it is a more mild presentation. Dual AAV vectors have delivered a 7.5 kilobase version, but it's still only half of the full length, which again is in the 11 to 12 kilobase range. And in animal models, it's been shown that full-length dystrophin provides a full functional restoration. But due to the techniques used in these studies, these are for animals only. The clinical application could be quite difficult. And so with Bobcat mRNA, it's a simple linear mRNA, and it's the first time that a full-length dystrophin can be delivered as such a linear mRNA. And that opens the possibilities of using this along with any other technologies that are developed for mRNA for these treatments. One issue that is known for mRNA is its transience, its short duration, which is up to a few days in vivo. However, it doesn't mean that dystrophin protein has a short half-life. In mice, full-length dystrophin's proteins half-life in cardiac muscle was up to 6 months versus 5 to 7 days for a microdystrophin. And that is a fact that's been confirmed by multiple additional studies. So even if the mRNA is short-lived, the DMD protein stays functional for months and months, meaning that the treatment can be quite infrequent. And we are looking into two methods for delivery. One is local and intramuscularly, and the other is systemic RNA delivery. And local and intramuscular is the one that could be applied now, and it's the one that we are actively developing. And it is a method that, if you remember, for mRNA has now been tested billions of times with a good safety profile. Compared to AAV gene therapies, AAV is one-time treatment and only once, and you can't take it back if there are adverse events or side effects, and it excludes those with pre-existing antibodies, requires immunosuppression, and is not redosable. There have also been several prominent cases of adverse effects over the years. And so in the case that it's not permanent or durable over time, a new approach is necessary. And in comparison, an mRNA approach is safer in that it can be stopped at any time, is infinitely redosable, and has a known good safety profile. And in addition, does not have to contend with pre-existing antibodies. Compared to existing exon-skipping approaches, where they deliver antisense oligos systemically to skip the exon for the mutation for the person with DMD has, a local full-length dystrophin can target all of the wild-type exons at once. So there's no need to develop a therapy for each one, which is currently the case. Each exon at most represents up to 12 to 15% of the patient population at most. And so there are many that are not served at the moment. And finally, neither approach delivers full-length dystrophin. Systemic RNA delivery for the full-length dystrophin is also in progress for us internally, but the local administration's ready today. And so we're currently closing in on assembling preclinical data for an IND. In terms of how it can be used, we believe that this is a complementary therapy to the existing methods, and we are seeking to treat non-ambulatory patients first.
Excellent, excellent. Thank you for the detailed explanation of not only the regarding delivery, but also just the what and the why behind it. speaking of preclinical data, I want to talk a little bit about the preclinical data that Elixirgen presented in March at the 2024 Muscular Dystrophy Association, or MDA, Clinical and Scientific Conference. A lot of good information came out of that, and I was hoping you could talk to us a little bit briefly about the data that you shared.
Yes. At this conference, we showed a preclinical proof of concept for this full-length dystrophin expression using Bobcat mRNA. And so this was first expression in vitro, but then also expression in vivo. And both of these are important data points for development for a therapeutic for DMD. And so particularly for in vivo, mouse skeletal muscle was injected with Bobcat mRNA expressing DMD, and we were able to see proper localization of the full-length dystrophin in vivo. In addition, we also shared functional data, and we used D2-MDX mice, which are a DMD disease model mouse that show a more severe phenotype than the standard MDX model that is used for much of the research today. And we shared two findings with this. So the first is that in these D2-MDX mice, repeated dosings of the Bobcat mRNA for the full-length dystrophin restored muscle strength for the disease model mouse to wild-type levels. And in comparison, the untreated, of course, had a deterioration over time. And in addition to this, for a single dosing, we showed a sustained improvement of muscle strength for up to 1 month with one single injection of this, again, Bobcat mRNA expressing a full-length dystrophin. And so not only have we seen some in vitro proof, but also some functional data for this Bobcat mRNA for DMD. And the test that was done was peak muscle strength using a grip strength meter, which is one of the ways that such mice are assessed. In the future, we're continuing to generate additional data in this way and other data that is considered standard for DMD treatments that are entering the clinic.
Amazing. Amazing. All right. Well, we'll keep our eyes out for what's next to come, certainly. But from your perspective, what is next on sort of the near-term future here for Elixirgen Therapeutics?
Yes. So we are actively seeking a co-development and out-licensing partner for Bobcat mRNA for DMD. We are an RNA company, and we are learning from the DMD community. We have been in touch with many of the prominent patient advocacy groups and have incorporated their initial feedback into the design for Bobcat mRNA. But the other component that's important is the experience of others with developing such therapeutics for the clinic and for people. So we are seeking this co-development and out-licensing partner in that way, and also for Bobcat mRNA's potential to be used in diseases other than DMD is also being actively investigated. We are also seeking a Series B financing for furthering this program and existing programs.
Good. Okay. Well, hopefully I'm sure you will receive the partnership opportunities you're looking for. That's fantastic. And certainly hope to have you back on sometime soon to kind of give us an update on both Bobcat mRNA and just what's been going on at Elixirgen since this conversation.
Thank you, Erin. I appreciate it.
Good, good. All right, listeners, that brings us to the end of our episode featuring Elixirgen's Aki Ko. Be sure to subscribe to the podcast and visit us at cellandgene.com, and we'll talk to you soon.